Kia ora everyone. This is my overview of the cells topic, life processes at the cellular level. So to start with, you do need to know the structure of some things in the cells. There's lots of organelles, but the ones you need to know about are the membrane, the structure of mitochondria, chloroplasts, a bit about enzymes and DNA structure. The whole topic covers these main things. There's three processes: photosynthesis, respiration, and the cell cycle, which includes DNA replication. And then there's information about transport and about how enzymes work, and they're important because they relate to all these things here. So three processes, but often what they'll do in questions is they'll link your knowledge about these things into one of those processes. So there's sort of five main bits, really. I'll turn the page. Trickiest part of the whole thing, sorry about that. Okay, so first part, transport. So in transport, you need to know about the structure of the membrane. So it's a double layered phospholipids that forms a structure, and through it there are proteins that can help transport things in and out. So the protein creates a barrier between the outside of the cell and the inside and controls what goes in and out of it. The main thing about transport is there's passive forms and active forms. The key to remember is the passive forms don't need energy, so anything that affects respiration will not matter, but the active forms do require energy, so the amount of respiration is going to make a difference here because ATP is required for the active forms. So I'm just going to run through these things on my little whiteboard over here. Sorry, it's a bit messy. So this is a cell. So the first form of passive transport is diffusion, and diffusion is just things moving from across concentration gradients. And so here's some, I'll zoom out a bit. I'm going to say this is some sort of chemical that's inside the cell. We've got a couple of these blue things, and we've got quite a few blue things outside the cell. So diffusion will happen without energy because molecules will flow what we call down the concentration gradient. They'll go from an area of high concentration to an area of low concentration just by themselves, and if the thing that's being transported is small enough, it'll just go through the gaps in the plasma membrane until the concentrations end up being equal, and then diffusion will stop. You might have another sort of molecule where the concentration is high in the cell. I've got lots of green in the cell and not so many outside the cell. So in this case, diffusion will happen, but in the other direction. So it'll go from an area of high concentration to an area of low concentration by diffusion with no energy required. Now, facilitated diffusion is similar, except in this case, we have a channel in the bilayer to allow something that's a bit bigger to go through. So it's facilitated diffusion, sorry, the scale's not very good, but we'll say this is a bigger molecule of glucose rather than, say, oxygen or carbon dioxide, and it needs a bigger space to travel through into the cell or out of the cell. Still not requiring any energy, it's just allowing the space for it to go through. So facilitated diffusion is just facilitating it by providing that channel. It's not using energy. And then the last one of passive diffusion is osmosis. So for this, osmosis is just diffusion, but it's the diffusion of water. So I'll go back to using my blue ones, and we're going to make the white fairy stoned water. Okay, so here we've got quite a lot of blue out here and not so much blue in here. If there's not so much of something, then there's going to be relatively more water in there. So we're going to say there's a high concentration of water here, and then when there's lots of blue out here, there's going to be less water. That's just sort of how it works. You can just look at the water concentration. It's a bit easier, but okay, so in this situation, we've got lots of water in here, not much water out there. So it's just the same as diffusion. The water is going to flow from an area of high water concentration to an area of low water concentration passively. And just here's a special name of osmosis because the movement of water and balancing out things is quite important in living things. Okay, we're on to active transport. So active transport is anything where we're going the other way. So we're going against the concentration gradient. Get rid of all that water. So now we're going to say that we've got quite a lot of stuff outside the cell, not much in, and we're going to go from low concentration to high. And so in order to do that, to go into where there's lots of stuff, you need energy to sort of push it into the area where there's lots. Okay, no energy required to flow down this way for diffusion, but active transport with things like ion pumps need energy to go into the area of high concentration. Often this is actually with something like, say, glucose, which relates to the topic because of respiration. Sometimes cells have quite a lot of glucose in them, and they can transport it passively or actively, but they use lots of it. So sometimes you have cells where they're actively pushing glucose into the cells. Anything that's called an ion pump, with the word pump in it, will be an active form of transport. And the last form of active transport is cytosis. And so that's where you have really big amounts of stuff coming into your cell. And this time a channel is not enough. What happens is the membrane actually forms around the big thing. It gets drawn into the cell, and then that bit of membrane joins onto the cell. That's endocytosis, going in, and the same going out. Here's our big bit of thing that needs to go out. That will sort of go out like that. It goes into there, and then that ends up over here with this bit of membrane around it. So membrane joins onto the cell or is taken off of the cell, and big chunks of membrane moving just uses energy. That's how it goes. So transport's all about concentration gradients, knowing the different forms. The size of the particles makes a difference, and also cell size and surface area are important. So cells don't get very big because they need to be able to diffuse things from the outside right into the middle of the cell, and importantly transport wastes from the middle of the cell out as well. And so if you have a much bigger cell, then it's just too far and takes far too long for things to diffuse all the way to the middle and all the way out again. And I'm going to carry on because we're already at 7 minutes and I've just done transport. Okay, enzymes. Enzymes are really important. They are catalysts that speed up reactions, and they do this because they have a specific shape, and they only do one kind of reaction. So you have lots of different enzymes doing different reactions. And so here's our enzyme. Okay, it's a protein, it's folded into a certain shape, and it has an active site. So one part of it's over here is going to be the active site, and it's going to either join two things together, or it might break a couple of things apart. So the substrate starts together and gets split apart. So they each do one part of a reaction, or one reaction, sorry, to make, in a chain of reactions to make a final product. They will have optimal rates they can work at, and so if the pH and the temperature and the conditions are all good, then, and they've got lots and lots of substrate, then they can work as fast as they possibly can. Sometimes they will denature, so if the temperature is too high or the pH is either too low or too high, that will change the bonds in the enzyme, and it will break some bonds, and that will change the shape of the enzyme, and once that shape is changed, then that substrate can't fit. So it's called denaturing because it makes it or not natural anymore. It's not its natural shape, irreversible, can't do its job. Other factors that affect the rate of enzymes working are cofactors and inhibitors. And so a cofactor is something that has to fit into the active site. So a little additional extra thing that fits in there and helps that reaction to happen. Inhibitors are the opposite. They stop things happening, and so an inhibitor can either compete, it can be a competitive inhibitor and compete with the substrate for the active site, and it goes in there, sorry about that, big truck going by, and it goes in place of the substrate in the active site, or it can be a non-competitive inhibitor where it actually attaches somewhere else on the enzyme, but attaching there changes the shape of the active site so the substrate can't fit. So they're slightly different to each other. We talked a little bit about pH and temperature. So low temperature will mean that things just can't happen as fast, but the enzyme is not denatured. Very high temperatures will denature the enzyme. pH too low or too high will break the bonds and the enzyme change its shape and denature it. The amount of substrate will matter, because if you only have a little bit of substrate, you can only do reactions at the rate of the, it'll sort of run out of substrate. But if you give it more and more substrate, it can go faster and faster until it gets to the point where it's saturated and it can only go at its maximal rate. It can't go any faster because you've given enough substrate. The overall amount of rate of reaction will go higher if you add more enzymes, of course, but eventually when you have the most enzymes and the most substrate, that will be the fastest it can go, and that's called the optimum rate. If the conditions are perfect, enzymes can work as fast as they can, there's lots of them, there's plenty of substrate, then it will all work at the optimum super fast rate. Great, okay, enzymes done. On to photosynthesis. Okay, might break this down. So, don't get overwhelmed with all the things for photosynthesis and respiration. Just think back to what's the main purpose of it. The purpose of photosynthesis is to make glucose, okay? It happens in plants in the chloroplast, so you do need to know a bit about the structure of a chloroplast. And what happens is the first stage is the light-dependent stage. Okay, we have these little stacks of green pancakes. Each pancake has got these things called thylakoid membranes, and it's in there that the green chlorophyll pigment is. And what happens is sunlight, the energy from sunlight hits the chlorophyll, excites some electrons, gives them some energy, and that energy is used to break water with its hydrogens and oxygens into its separate parts. It breaks into oxygen, and that disappears off as a waste product. And then they've got hydrogen left, and the hydrogen gets carried to the next stage. The next stage is the light independent stage, doesn't need the sun anymore. And what happens, it happens in the stroma, so in the liquidy part in between those stacks of grana. And that hydrogen from the breaking down of the water is joined onto carbon dioxide that the leaf has got in through the holes in the bottom of it. And those two things joined together can make glucose. So we've got our hydrogens and our carbons and our oxygens, and that's the formula for glucose. If you forget all the steps, just remember the overall equation. Remember, we're trying to make glucose. We know that plants take in carbon dioxide and they get rid of oxygen, and we have a bit of water in there as well. So when you write it, you're going to write the words or the symbols, and you have to make sure that you put chlorophyll and solar energy or light over or under the arrow, because those things are not used up in they're not changed in the reaction. If you don't know how to do the balance formula, the easy way to do it is just look at, you've got your great big glucose here with all your letters, so we're going to ignore that one, but the smaller ones, the oxygen, the water and the carbon dioxide, you're just going to put a great big 6 in front of all of them. They all have the same number in front, so it makes it nice and easy. So that's your balanced formula. And so you can just talk about the fact that carbon dioxide goes in, water goes in, these things do that, makes this, if you forget the individual steps and the names of them. And then finally, what affects the rate of reaction for photosynthesis? Well, think about, what do you need? You need carbon dioxide, you need water. So if the plant can't get enough water, then that's going to affect the rate of photosynthesis. It needs sunlight. It's also going to need enzymes to do all these things. And so enzymes depend on the temperature. So if it's colder, that won't work as fast. It's also, enzymes might denature. And there might be inhibitors around that might stop the enzymes working, because remember, these are all chemical reactions making this, and so anything that affects an enzyme can affect photosynthesis. And I've said already the substrates going into the reaction as well, and that is photosynthesis. On to respiration. Okay, so again, remember what the big overall idea is. We're trying to make energy, and we're making energy from the glucose. It happens in organisms all the time, and we use it, lots of it, for active transport in our cells, for movement, and for making molecules, proteins and things. You need to know the structure of the mitochondria, and remember that, in fact, the very first step happens in the cytoplasm of the cell before it even gets into the mitochondria. And you need to know the difference between aerobic and anaerobic respiration. So aerobic is with oxygen. And the first step is called glycolysis. Again, if you don't remember the name of it, don't worry, just describe what happens. So we've got our glucose, and it's broken in half to 2 pyruvate molecules. So pyruvate's a funny name, isn't it? But it's just basically splitting the glucose in half. So we've gone from C6H12O6, and we go to C3H4O3. 3. That doesn't really matter, you don't have to remember that, but it's just makes you realise what's happening with the reaction. So it's just being split into 2 bits, and that actually creates 2 molecules of ATP just in that stage. Then we go into the Krebs cycle. Here that pyruvate molecule is broken into carbon dioxide and water. The carbon dioxide this time is the waste product, and it disappears out of the mitochondria, and then the hydrogen gets taken to the 3rd stage, which is called the electron transfer chain. Here the electron from the hydrogen is used, the energy from that electron that's taken off the hydrogen bounces along a little chain and makes 36 ATP. So lots and lots of ATP are made in this final stage. Then the electrons return to its hydrogen. Hydrogen joins on to the oxygen and makes water. And so you can sort of start to see that this is like the opposite way round to photosynthesis. The overall equation is glucose